My name is Dan Cook. I thank the FAIR and ASA conference organizers for the opportunity to discuss my research on the impact of renin receptors on presynaptic calcium regulation in hippocampal neurons. Communication between neurons within the central nervous system is accomplished primarily by the release of chemical neurotransmitters from nerve terminals. An action potential depolarizes presynaptic voltage-gated calcium channels. The resulting calcium influx into the cytosol causes conformational changes to synaptic proteins that fuse synaptic vesicles with the cellular plasma membrane, releasing neurotransmitter into the synaptic cleft. One organelle often overlooked in this model is the endoplasmic reticulum. As highlighted by this 3D reconstruction of focused ion beam electron microscopy of an axon, the ER, in green, extends throughout the length of an axon and is closely associated with synaptic vesicles, shown in blue, at nerve terminals. In sarcoplasmic reticulum, the analogous structure of myocytes, the SR has a role in releasing calcium in response to calcium influx into the cytoplasm, a process called calcium-induced calcium release that leads to muscle contraction. Calcium-triggered efflux of calcium stores from the endo or sarcoplasmic reticulum is mediated by ryanodine receptors, which are intracellular calcium channels spanning the ER membrane. All three forms of ryanodine receptors are expressed in the central nervous system. Previously in the Ryan lab, a genetically encoded fluorescent indicator of calcium was targeted to the endoplasmic reticulum to study resting levels and dynamic calcium fluxes in this compartment. Surprisingly, field depolarization of neurons caused a net increase, not decrease, in calcium levels in the ER hippocampal neurons. Thus, ER calcium stores seem to have a different role in axons of central nervous system neurons compared to the better understood model in muscle cells. This finding raises the question, what is the role of ryanodine receptors in neurons of the central nervous system? The answer to this question may not only address poorly understood biology underlying synaptic communication, but may also be relevant to the pharmacology of commonly used drugs that impact ryanodine receptors, such as volatile anesthetics and local anesthetics. This question was approached using primary dissociated hippocampal neurons from postnatal day 0-1 rats, cultured onto cover slips, transected with fluorescent biosensors of synaptic markers, loaded onto a custom perfusion chamber capable of delivering a field potential to depolarize the cell, and studied with fluorescence microscopy. For imaging, cells were maintained at 37 degrees Celsius and perfused with thyroid solution containing 1.2 millimolar calcium, the physiologic concentration within the cerebral spinal fluid. Co-expression of the synaptic marker mRuby-synapsin with the cytosolic genetically encoded calcium indicator GCAM6 allows calcium levels and fluxes to be precisely measured at individual nerve terminals. For visualization purposes, these ROIs highlight only a few of the synapses present in the field of view that were analyzed. Field depolarization across the cover slip allows evoked changes in calcium to be quantified. These images show the baseline subtracted peak response in calcium influx in control and after a 30-minute treatment with the drug ryanodine at a dose known to potentiate the receptor. Ryanodine receptor potentiation caused a net decrease in evoked calcium with the mean inhibition of approximately 38% compared to control conditions that the neurons studied. However, the mean response obscures interesting synaptic variability in the response to ryanodine receptor potentiation. Each dot on this plot represents the response net calcium influx of a single baton before and after ryanodine treatment. The dashed line is a slope of 1, such that data points to the right of this line represent inhibition calcium influx by ryanodine treatment, and points to the left represent an increase in calcium influx. This trend of substantial synaptic variability was evident in all the neurons studied. One interesting phenotypic subset is a set of batons in which a demonstrable calcium influx was abolished by ryanodine treatment. By setting a threshold of 1 standard deviation of pre-stimulus variability above the baseline fluorescence, we identified a subset of batons in which calcium influx was silenced by ryanodine treatment. Ryanodine treatment also caused in a subset of batons a sustained increase in cytosolic calcium or spontaneous calcium fluctuations, often referred to as sparks. Under control conditions shown here, there is little variability in resting calcium in the neuron. However, pharmacologic potentiation of the ryanodine receptor led to sparks, which are distributed heterogeneously in space and time. This figure is a common graph of the net increase in calcium after ryanodine treatment compared to control conditions across all batons imaged in this field. Setting an arbitrary threshold of 5 standard deviations above the mean fluorescence value control conditions, and looking at this region of the common graph, you can better appreciate the stochastic nature of calcium sparks. A mean A mean of 37% of Bhutans crossed this threshold, showing either a brief or sustained increase in cytosolic calcium due to ryanodine receptor potentiation. These studies demonstrate that ryanodine receptor potentiation causes, on average, a decrease in cytoplasmic calcium influx in response to neuronal depolarization, but there is significant synaptic variability even within single neurons. A subset of Bhutans can have evoked calcium influx abolished by ryanodine receptor potentiation. We also observed calcium sparks that are heterogeneously distributed across Bhutans of a neuron in both space and time. That potentiation of ryanodine receptors, a channel allowing calcium efflux of ER calcium stores into the cytosol, could decrease EVO cytosolic calcium responses, suggest that there is a presence of a feedback system of ryanodine receptors on either voltage-gated calcium channels or perhaps the active potential propagation. Thus, these data indicate ryanodine receptors could have a significant impact on synaptic function in neurons of the central nervous system, which has relevance to diseases and drugs that impact ryanodine receptors. I would again like to thank FAIR for the generous support of this research and the opportunity to present, acknowledge my mentors and other funding sources. Thank you for your attention.